Right, as far as archaea, we can do the same thing, but much quicker. With archaea, do know that they do have a cell wall, but it's different. So with archaea, it's even less details that you need to worry about. With archaea, first of all, there is an exception that we talked about earlier. They do have, instead of peptidoglycan, they have pseudo-peptidoglycan. They have a different form of peptidoglycan. It has beta-1,3, not beta-1,4. In fact, don't worry about the details of it because they can get a little bit. Just know instead of PG, they have pseudo-peptidoglycan. Don't worry about the differences between this and that. But also, they can, if they don't have pseudo-peptidoglycan, what would they have? Either they don't have the cell wall, or they have pseudo-peptidoglycan as a cell wall, or they have the, I think, the S layer, exactly. Or they have the S layer. Those that have pseudo-peptidoglycan, they stain gram positive. Those that have the S layer stain gram negative. That's archaeal cell walls. Cell membrane. Again, try to flowchart. And I'll spend a little bit more time on the old material because it's old, I guess. And it's just, the new material is frankly less complicated. But anyway, cell membrane, same thing. Bacteria, archaea. Bacteria, I mean, let's just put words that, obviously they're both lipid-based for the most part. With bacteria, you have ester linkages. You have hopenoids. These are the sterile equivalents. And unbranched lipids. Right? These are the things that come to mind as far as, with archaea, they're different. With archaea, you have ether linkages. And hopenoid, you have basically isoprenes. And you have branched lipids. So basically, if this is, essentially, they look like this. Here, it's not really branching, but if anything, they look like this. That's a big difference, right? It's the same line that just twists. Here, it's one line, and there's branches coming out of it. With ether, you have two types, right? We have diethers, and we have tetraethers. Which of these is the much more stable membrane? The di or the tetra? It's actually, if it was di, basically this is a diether, right? You still have two tails, so you would need another two tails to come to form the membrane. With a tetra, it's just one big block, right? It's just one big block, so this is obviously much more stable. Because if you have a diether, it's a regular phospholipid basically, and it can easily twist and turn. This is much more stable. This is what is found in usually thermophilic organisms. Questions about? Notice I did not mention sterols. Sterols are very rare. Sterols are largely in the context of mycoplasma. Hopenoids are what's questions about with the molecular portion, like plasmids and nucleoid, we'll discuss them in more detail in the exam 3 material, which is 7, 8, and 9, so I'll leave them for now. Pili, inclusion bodies, anything with inclusion bodies. Here you don't have to go into details again, but so for example, we talked about magnetosomes, right? Just obviously they can don't know the organisms that produce them, know that you can have magnetosomes that allow an organism to orient itself with respect to a magnetic field, right? What are gas vesicles or vesicles used for?
Transport gas.
To help
The flotation.
For flotation devices, right?
Okay.
Gas, they fill up, floating up and down, right? True or false, that could be a true and false question. Bacteria have a cytoskeleton. True, right? It's not just eukaryotes, they have a cytoskeleton in the true sense of the word. Pili, let's do the same thing, divide. Right, we talked about common, sex. Common are also called fimbriae. Sex are called fertility. Here again, notice the compare and contrast, not high and not actual details. These are many, these are few. These are short, these are long. What is one word that links both of them? What is the function, in other words? What do pili function in? One.
Attachment?
Right, both of them function in attachment. What's the difference? These attach to cells other than themselves, or to rocks, or to whatever. These attach to themselves. That is what's used in that's what's used in what do bacteria use sex fertility factors for? Conjugation, right? This is chapter 9, conjugation. Flagella, same idea, right? First, know the terminology. So, for example, if you have a bacterium that looks like this, these being the flagella, what would this be called? Right, you have flagellum on either side. These are amphitrichus, right? Opposite, amphi. If it has something like this, it's slowful. If it has something like this, what would that be? Petty, all over, right? Do know the parts of the flagella, right? So, and this is a very long structure. Again, it's going to be nicer if I do show it. This is the weirdest flagellum I've drawn. Okay. Something like that, right? Filament, hook, basal body, right? You have the L, P, S, N, and the C ring, right? All of them are found in gram negative. Only the bottom two are found in gram positive, right? That's pretty much it. Motility, chemotaxis, very briefly, remember they run and tumble, right? When they run, the flagella are rotating counterclockwise. That allows them to go in a straight line. Then they tumble, the flagellar bundle falls apart, and then they go into another. So they run, they tumble, they run, they tumble, they run all the way towards their surface. Questions about... There are other forms of motility, so remember them, right? So for example, the swarming bacterium is Proteus, right? The gliding bacterium, we only discussed one, Simonsiella. Endospores is another, at least one question on endospores. Be able to, so for example, it's largely gram-positive, right? And they're soil organisms. That's one thing to keep in mind. Bacillus, Clostridium are the two medically important genera. The cell that's producing the spore is called the Well, the vegetative cell is the actively growing spore, right? But it doesn't have to. So the vegetative cell that produces a spore is called the sporangia. They're heat resistant. What makes them heat resistant? Several layers, and the chemical calcium dipicolinic acid. So calcium dipicolinate or dipicolinic acid, same thing. Streptomyces produces ectospores, exospores. These are the ones that look like fungi, right? These are not as heat resistant. And there's a third one that we discussed, which is produced by Azotobacter. These are cysts. Don't worry about Anabaena. There's one about Anabaena and cyanobacteria. And actinides, don't worry about that portion. Questions about 3. 5, I'm sorry, 5, you can say 5. The chemotherapy, which is 27, here again, I'm not going to go into details as to, so for example, well, certainly with protein synthesis. I'm not going to ask you, is it the 30S or the 50S? I'm going to ask you, is it protein synthesis? Is it cell membrane synthesis? Is it cell wall inhibition? And even with cell wall, don't break it down further, right? So for example, like with bacitracin, don't worry about whether it's the recycling of the carrier or the cross-linking. Just know it's a cell wall inhibitor. Does that make sense? So because there was the cell wall inhibitors, they could be preventing cross-linking, preventing transglycosylation, preventing the recycling of the carrier, etc. Just know them as one big group, which is a cell wall inhibitor. Very quickly, we talked about the 3 big producers, right? So that's could be one question right there. The number 1 producers are the actinomycetes. The number 2 producers are the molds, largely penicillium. Here it's streptomyces. Here it is largely penicillium. 3 is bacteria other than 1. So, right, and then bacillus is the big one. Narrow versus broad spectrum. So narrow versus broad spectrum. That's another thing to know. Static versus cidal, right? Static versus cidal. Selective toxicity, right? That's the idea of the magic bullet. You want a bacterium to be harmed and the host not to. So beyond that, as I mentioned, there's probably going to be a total of probably about 8 questions on antibiotics, roughly. So know those, whatever, 20, 25 antibiotics. But the questions would be very similar to ones that I've asked earlier. I will give you 5, for example. Let's do one. Which of these antibiotics does not fit? And again, the exam is in 4 days, so I don't expect all of you to be ready now to answer this. But my job is to give you examples, and this is what I'm doing. Actually, my job is beyond that a little bit, but anyway. Okay, so tetracycline, streptomycin, penicillin, amphotericin, it's probably a little bit harder than, but you have 5 antibiotics. Which of these does not fit with the others? Now there has to be a rhyme or reason for the other. You start with this. This is a bacterial antibiotic against protein synthesis, right? This is a protein synthesis inhibitor. Same thing with this. So these fit together. Penicillin is a cell wall inhibitor, right? So is bacitracin. So you have two cell wall inhibitors. This looks a little bit out of place. What is terbinafine? Terbinafine is the only antifungal of the list. Terbinafine is Lamisil. It's the only antifungal. All others are antibacterial. You could have the same thing with 4 antifungals, 1 antibacterial. So my point is, know them directly and be able to differentiate. So this is as detailed a question as I can ask. More likely, most of the others would be much more straightforward. Which of these 5 targets fungi? Or which of these 5 targets bacteria? Which of these, whatever. Right? Now there will be some questions where you have more than one choice. That is true. Right? So it'll be 3 and then 2 of the above or all of those. There will be some questions like that. But if you know the material, you know the material. So again, no cell wall inhibitors in the context of cell wall. Don't break it down, like bactoprenol inhibitor or whatever. With DNA and RNA, you have to be able to differentiate, right? So with DNA, in fact, we can go over them real quick. Cell wall inhibitors, right? So start with cell wall inhibitors. Don't worry about beta-lactams even. Just be able to list them all. Cell membrane. Right? Cell membrane inhibitors, right? Or disruptors in this case. Polymyxin, for example. That's the big one. Here you have the beta-lactams, etc. Then we talked about protein synthesis inhibitors. Right, here you have tetracycline, streptomycin, chloramphenicol, oxazolidinones, a bunch of them. DNA and RNA, be able to distinguish between them, right? Here we talked about the quinolones, right? Metronidazole. With RNA, rifampin, actinomycin D. The anti-metabolites too. So, for example, I know what do sulfa drugs do. What does trimethoprim do? Right, sulfa is the dihydroteroate synthetase. This is the reductase. These are the 2 enzymes. Right. Isoniazid, narrow spectrum against tuberculosis. Dapsone against leprosy. Same thing with the antifungals. Be able to, if you're dealing with the antifungals, again, with the antifungals, there's relative, well, certainly much fewer than these. But quickly, with antifungals, there is a big difference between cell membrane disruptors and inhibitors, that is. With fungi, there is no inhibitors, they just disrupt. With fungi, with bacteria, that is, they do not inhibit, they just disrupt. With fungi, there are disruptors and inhibitors, right? So for example, amphotericin B and nystatin, these are the polyenes, they disrupt. The inhibitors are the azoles. Terbinafine is an example. Echinocandins, cell wall inhibitors. Microtubule assembly, rhiziofulvin. Pifluorocytosine, nucleic acid. Questions about okay, so with 7, 8, and 9, that's the last exam, and again the class did quite well on that one. So hopefully it's still fresh in your memories. So I'll go through them again, highlighting the most important stuff. Just from 7, remember that's the only reading assignment on the final exam, the part that is in the handout actually, the one that I go over nucleotide structure, DNA versus RNA, pyrimidine versus purine. So again, if you have, let's say, percentage of adenine, percentage of cytosine, percentage of guanine, and you're told, and this is an unknown sample, the percentage of adenine is 30, the percentage of cytosine is 30, the percentage of guanine is 30. And then here are your choices. D and E. A single-stranded DNA, single-stranded RNA, double-stranded DNA, double-stranded RNA, and can't tell. If you're given these three, which of these is it? It's an unknown molecule.
Can't tell.
You actually can't tell because this could be U or it could be T. You have to know this. Right? If I told you this is DNA, you're told that it's DNA. Right? So now can't tell. Actually, now you're down to these two. So it's true and false. Is this double or is this single-stranded DNA? Sorry? This would have to be single. How can you tell? Well, if this is 30, this is 30, this is 30, what's left is 10. 10% T. A's and T's are not equal. It's got to be single-strand. Purines are adenines and guanines. Cytosine thymines are pyrimidines. The rest of the straightforward. I mean, the rest is pretty straightforward. If you have two samples and you're comparing which, or two unknown molecule or known molecules, comparing which is more stable, you look for more G's and C's, right? Because it's more triple bonded, it's stronger. As far as the polymerases, don't worry about the DNA polymerases in eukaryotes. We'll address the ones in prokaryotes, okay, bacteria. They all polymerize 5 to 3. That's the main. You will get questions of putting things in order. So, for example, well, let's start from the beginning. The origin is called ODC, right? It's rich A's and T's. You have one terminus. DNA A bind first. They open the helix. Helicase opens it further. You put primase. DNA pol 3 comes in. Right? What's the role of DNA pol 1? To remove the primers. Replaces them with DNA. Again, you're not gonna. Don't worry about the leading and lagging orientations, 5 and 3, etc. So, but do know like what are Okazaki fragments. You will not get labeling diagrams on this one. Same thing with termination. Don't worry about the process of decatenation, etc. Again, be able to put these proteins in order and not worry about the details. So, for example, I mean, other questions would be, yeah, so don't worry about my eukaryotic DNA replication. Right, so any questions about, so remember, pol 3 is the big one, pol 1 is the removal of the primers, pol 2 is repair. So that's the take-home message really from replication. Transcription is a little bit more nuanced. Again, go back to the basics. We have the RNA pol holoenzyme, right? At least in E. coli, that's what we're focusing on, the holoenzyme, the big one. It can be 2 parts, sigma, or actually it's, well, sigma factor and core. The core is alpha 2, beta, beta prime, and in many cases omega. That's why I'm putting it in parentheses. Sigma factor is sigma factor. The role of sigma factor is to bind to the promoter. After it binds to the promoter and transcription initiates, it's left, it's gone. Sigma factor continues to the end. How does it know where to end? Row dependent or row independent. You don't have to go beyond that. So someone says row factor, transcription termination in bacteria. Someone says sigma factor, transcription initiation in bacteria. Right? Again, don't worry about the -10 and the -35 sequences. But do know, for example, true or false, the promoter is transcribed. True or false? False. The promoter is not transcribed. The promoter is there, so the transcription starts downstream at the +1 site. The promoter is not transcribed. The promoter is not transcribed. Right? You have to be able to tell differences. So, for example, right? let's say very quickly, A, C, C, G, G, T, T, let's make it easy. T, T, G, G, C, C, A. Actually, that came out to be a right? Am I correct? Yeah, correct. Okay, so 5, 3, 3, 5. And you're told that RNA is going this way. True or false? RNA is going this way. This is the coding strand, true or false? RNA polymerase is coming this way. Is this the coding strand?
Yes.
True. How? If this is transcription, that means this is the template. How can I tell? Well, this is my 3' end. So the 5' end has to come out first, which means this is now the protein, right? True, the coding strand is always the strand that goes 5 to 3. No, it is true, right? It's always the strand that goes 5 to 3, but you don't know which is which. It could be 5 to 3 this way, but it's always going 5 to 3. But it's not always the same one. Does that make sense? It's not always this one, but it's always the one that's going to go this way. This one is the coding, and now the gene is going this way. Right? The genetic code. And then remember, with nucleic acid chemistry, you really worry about two things. If you want to put the DNA and an RNA together, or RNA and RNA, or DNA and DNA, whatever, okay? You only worry about two things. They have to be complementary and anti-parallel. So if you're given two sequences and asked whether they're complementary or not, rule number one, you obviously have to ensure that they're A and U, C and G, you know, A and T, etc., complement. And the other one, they have to be an anti-parallel. So don't automatically assume that, well, you know, that's going to be the one. You have to match the 5 to the 3, the 3 to the 5, right? So keep that in mind. The genetic code, at least one question on the code. Very quickly, you know, it's RNA, not, it's usually RNA, not, never DNA. Well, let's go back. It's usually in RNA. If it's in DNA, it's the coding strand. So you only replace these with U's. That's the only thing you need to worry about. It's a triplet code. It's degenerate. It is not overlapping. It is commaless. It is nearly universal. The exception, do know the same codons that we talked about earlier. AUG is the start. The UAG, UGA, UAA are the stop. Methionine is AUG. N-formal methionine starts bacteria. Methionine starts eukaryotic and archaeal. Questions about the code? With translation, same thing. I'm not going to ask the details of the. So let's go back. Do the same thing. We'll start and then go through some examples. So first of all, what are the ingredients, right? You have to know it's obviously messenger RNA. Transfer RNA is involved. Ribosomal RNA is involved. How are these all involved? Well, this one carries the message, right? This one brings the amino acids. And this one makes up the ribosome. Ribosome, you have a small and a large subunit. Be able to tell the differences or differentiate between bacteria, eukarya, etc. Small in bacteria is 30, 50. They combine to give you 70. Now, let's go back to transfer RNA. Again, remember the structure, right, of transfer RNA. And remember that the one that is longer is the 3' end. That's the one where the amino acid goes. This is the 5' end. The enzyme that charges it, aminoacyl tRNA synthase. Aminoacyl tRNA synthetic. Now, as far as the steps of translation, just as I asked earlier,